Hello and good morning, everybody. Welcome to GemPharmatech's monthly webinar series. My name is John Jackson. I'm part of the marketing team at GemPharmatech. And today we'll hear from Dr. Wenzeng, our Senior Study Director for our autoimmune pipeline, as he goes over the webinar topic, Rethinking Asthma Preclinical Models, Matching Mouse Models to Drug Mechanism Across Asthma Phenotypes. If you don't know GemPharmatech, I'm only going to spend a brief minute or so explaining who we are. Our mission is to engineer a healthier world by pushing the boundaries of biomedical innovation. What does that mean? It means we provide the models, the data, and the insights that move drug discovery or research forward. We have the biggest library of proprietary in vivo models that fuels these preclinical solutions. We do this work globally. We help scientists, whether it's in academia or industry, complete over 1,000 preclinical studies each year. And in terms of model generation, we have the capacity to generate 6,000 new strains each year. Dr. Wenzeng is going to speak about this webinar. It's obviously related to asthma. However, we do work across all disease indications. So if you are interested in other topics, feel free to reach out to us at sales@gempharmatech.com. At the conclusion of his slides today, there will be a brief Q&A session. We do these monthly, so if you follow us on LinkedIn, if you're subscribed to our newsletter, you'll certainly get updates about the next one next month. With that, I will leave the rest of this to Dr. Wenzeng and enjoy his talk.
Okay, hello everyone. I'm Wenzhen from GemPharmatech. Welcome to our webinar today. Today we are going to talk about rethinking asthma preclinical models, matching mouse models to drug mechanism across mouse asthma phenotypes. And to give an overview of today's session, we have structured the presentation into 3 chapters. First, we will look at the shifting landscape of asthma drug development. We will bridge the gap between clinical insights, specifically the type 2 and non-type 2 endotypes, and how they translate into our animal models. Second, we will discuss benchmarking success. Here we will analyze the classic eosinophilic paradigm and what we call the dupilumab wall, a phenomenon where high clinical efficacy sets an incredibly high bar for new drug candidates. Finally, and perhaps most importantly, we will explore the territory beyond the eosinophilic paradigm. We will dive into our latest data on mixed granulocytic and neutrophil dominant models, which are essential for addressing the significant unmet needs in non-type 2 asthma. So now let's begin with part 1, the shifting landscape of asthma drug development. In this section, I will briefly recap the symptoms and the global burden of asthma, but our main focus will be on the transition from broad clinical symptoms to specific molecular endotypes. Understanding the bridge is the first step in selecting the right preclinical model for your specific program. So asthma is a chronic inflammatory condition of the airways characterized by episodes of reversible airflow obstruction. As you can see on the left, the healthy airway is clear with relaxed, smooth muscles. In contrast, an asthmatic lung suffers from a triple threat: muscle tightening, significant airway swelling, and excessive mucus production that clogs the passages. Clinically, this manifests as the spectrum of symptoms from dry cough, night cough, dyspnea to wheezing. The epidemiology of asthma, we can find a global disease that affects near from 1% to 29% of the population across various countries. Asthma is a global health challenge. Currently, the global burden is highest among adolescents at 11%, followed by 9.1% among children and 6.6% among adults. An accurate diagnosis is the bridge to effective therapy. According to the latest JNA and Lancet frameworks, we evaluate the pretest probability of asthma based on the variable symptoms, those triggered by exercise, viruses, or allergens. versus constant monosymptomatic coughs, which may suggest alternative diagnosis. Beyond history, we rely on objective diagnostic tests, spirometry with reversibility, peak flow variability, and bronchial challenges. Crucial for our discussion today, we also look at type 2 biomarkers such as fractional exhaled nitric oxide levels and blood eosinophil counts to phenotype patients and predict their response to targeted biologics. The pathogenesis of asthma is far more complex than simple inflammation. It is a multiple faceted process of immune hyperresponsiveness and the structural alternation. So the key drivers include, firstly, the airway remodeling, that's permanent structural changes, including subepithelial fibrosis. And second, the SM hypertrophy also results in the thickening of airway smooth muscle, which increases contractile forces. And third, the mucus hypersecretion lead to the luminoplex we saw earlier, and the sensory nerve dysfunction is contributing to the heightened cough reflex and hyperresponsiveness. And type 2 mucosal inflammation is traditionally driven by the interleukin 4, 5, and 13 axis, though we are increasingly recognizing the role of non-type 2 pathways involving neutrophils and interleukin 17. So a key point in asthma is that the heterogeneous mechanism and the phenotype exists in asthma patients. The classification of asthma largely determines the disease mechanism, immune response, as well as the drug development. So in asthma, it can be divided into type 2 and non-type 2 asthma. Type 2 asthma is dominated by eosinophils and the interleukin 4, 5, and 13 signaling pathways. These patients typically respond well to corticosteroids and the existing biologics like dupilumab. On the other hand, we face the challenge of non-type 2 asthma. This phenotype is often neutrophil dominant, driven by interleukin 17, interleukin 6, and TNF alpha. These patients are often insensitive to corticosteroids and currently have no approved biologics, representing a massive unmet medical need that we must address in the lab. So as the industry moves beyond the eosinophilic paradigm, we are seeing a surge in clinical candidates targeting the unmet need of non-type 2 asthma. Several key alarmin inhibitors, like the targets for interleukin 33 receptor and interleukin 33, have shown promising results in patient populations where traditional type 2 biologics struggle. So in the first row, we can find that the astegolimab demonstrate its potential as a tailor-made therapy for non-type 2 patients. Among patients with low baseline eosinophils, it reduced the annualized exacerbation rate by more than 50%, whereas the effect was significantly lower in high eosinophil populations. And for itepekimab, the candidate has shown a broad applicability. Phase 2 data indicates significant prevention of asthma loss of control in both eosinophil low and eosinophil high subgroups, suggesting its efficacy is not strictly dependent on type 2 high biomarkers. And in the third row, the tozorakimab, while it faced the challenges in meeting primary endpoints in some studies, high dose tozorakimab showed a clear trend in improving lung function for the non-type 2 population, particularly in severe cases with frequent exacerbations. So these clinical insights raise the unmet needs and requirements in bridging the gap between bench research and these emerging non-type 2 asthma clinical outcomes. So when it comes to preclinical research, one of the most critical factors is the model classification and responsiveness to standard of care treatments, specifically glucocorticoids and biologics like dupilumab. So at GemPharmatech, we also categorize our asthma model into two distinct groups based on their therapeutic response profiles. So the first category is glucocorticoid responsive asthma model. The most classic model is OAA-induced asthma model. This is a classic Th2 cell dominant model with high proportion of eosinophils. We can find the significant infiltration of eosinophil cells in the lung and blood of asthma model, while the administration of glucocorticoids like dexamethasone can significantly inhibit the eosinophils and exerted near perfect efficacy. But when it comes to glucocorticoid-resistant asthma model, we can find a completely different efficacy in dexamethasone treatment group. So the HDM plus LPS-induced asthma model is a representative asthma model, like glucocorticoid-resistant asthma model. In this model, it is a Th17 cell dominant characterized by neutrophilic infiltration, and we can find much more infiltration of neutrophils in lung, and we can find that after dexamethasone treatment, the efficacy in inhibiting eosinophils is impaired. So that's the most different part in the glucocorticoid resistant asthma models. So based on these results, now in GemPharmatech, we have developed four different asthma models. We can divide these asthma models by glucocorticoid resistance and the infiltration of inflammatory cells. So if we classify these models by glucocorticoid resistance, we can find that the classic OAA-induced asthma model, the HDM-induced asthma model, as well as alternaria alternata-induced model is the classic glucocorticoid responsive model because these models are the classic type 2 immunity related asthma models. So it means it will respond well to glucocorticoids, and the infiltrations and the inflammatory cells infiltrated in lung is mainly Th2 cells and eosinophils. And one little differences in this model is that in HDM asthma model, considering the characteristics of HDM, it can induce a whole activation of immune system. So it also involves in the participation of alarming. And for the alternaria, alternata-induced model, we can the differences is that it is induced and activated by the innate lymphocyte 2, while the OVA and HDM model is activated by the Th2 cells. So that's the characteristics of glucocorticoid responsive models. And for glucocorticoid resistance model, we have OVA plus chitin-induced asthma model and HDM plus LPS-induced asthma model. So in OVA plus chitin-induced asthma model, it is characterized by the infiltration of both eosinophil and neutrophil. So it exerted a mixed type of infiltration, and the responsiveness of glucocorticoids impairs in this model. And for HDM plus LPS model, as mentioned previously, it is a completely neutrophil dominant model by the infiltration of Th17 cells. So in this model, we can also find the impaired efficacy in glucocorticoids and dupilumab. So that's all about the first part, the shifting landscape of asthma drug development. Now I think we can step to the chapter 2, bring to our second chapter, the benchmarking success. So for decades, the eosinophilic or type 2 pathway has been the dominant paradigm in asthma research. This success culminated in the approval of biologics like dupilumab, which has revolutionized the patient care. However, this success creates a unique challenge in the lab when the current standard of care works too well in traditional models. How do we demonstrate the value of a next generation candidate? So in this chapter, we will examine the data behind the dupilumab wall and what it means for our efficacy studies. So firstly, I will quickly introduce the features of HDM-induced asthma model. Typically, this is the asthma model we applied when evaluating the dupilumab efficacy. So the immune features of HDM-induced asthma model is that it involves with the airway epithelial cells and to the cooperative responses of immune cells like innate immunity, like dendritic cells and the innate lymphoid cell too, and other basophils and mast cells. And finally, it will reach to the Th2 cell differentiation as well as B cell activation. So the modal characteristics, including the eosinophils dominant in lung and BAF, and with the Th2 inflammation in blood and lung. And we can also find the activation of B cells, where we can find the antibody secretion, including serum IgE levels and anti-HDM specific IgG levels. And we can also find the damage of lung by HDM modeling at histological level. So here is the study design of the dupilumab in HDM-induced asthma model. So to evaluate the efficacy of dupilumab, we need to use humanized mice. So here we applied the B6 background, humanized the interleukin-4, and the humanized the interleukin-4 receptor mice with HDM intranasal instillation. The whole study takes 28 days to complete in vivo studies. And now I will quickly go through the dupilumab efficacy data in HDM-induced asthma model. So firstly, we can find that on the study endpoint, 28 days after HDM modeling, we can find dupilumab treatment significantly inhibited the anti-HDM specific IgG level, the serum IgE level, as well as serum human interleukin-4 levels. So basically, we can find near perfect efficacy in these indexes in the dupilumab treatment group. And when it comes to the immunotyping, we can find the same results. The one of the most featured characteristics of HDM-induced asthma model is the eosinophils dominant type. So in here we can find in the lung we can find there is around 1.5 million eosinophils in lung, while the number of neutrophil is only around 50,000. So it means it is typically an eosinophil dominant model. And after dupilumab treatment, we can find the nearly perfect inhibition in eosinophils. And we can find the similar results in inhibiting circulating blood eosinophils in dupilumab treatment group. And when it comes to cytokines, we can still find the similar results. So when we collect the lung and extract mRNA for the cytokine quantification, we can also find significant inhibition in dupilumab treatment group in inhibiting CCL11, CCL17, interleukin 13, interleukin 33. So these indexes and chemokines are largely involved in the activation of type 2 immunity. And these results means the administration of dupilumab significantly inhibited the activation of type 2 immunity in mRNA level in the lung of mice. And also, we can find the similar results in the protein level. So we can find after dupilumab treatment, it significantly inhibited mouse interleukin 5, mouse interleukin 33, and mouse TARC compared to the modeling group, which means the dupilumab treatment also realized the completely inhibition in the type 2 immunity at protein level. And when it comes to pathology, basically we can find the same data. So in the modeling group, we can find the infiltration of inflammatory cells in the lung, as well as the thickening of lung epithelial cells. And after the dupilumab treatment, we can find the significant decrease in total asthma model scores. So when we make a brief summary of the data, of dupilumab in HDM-induced asthma model, we can find that after dupilumab treatment, we can find near perfect data in inhibiting serum IgE, HDM-specific IgG1, and human interleukin-4 levels. And then we can also find perfect data in inhibiting eosinophils in lung and blood, and also the perfect data in inhibiting cytokine release of lung in mRNA and protein levels. And we can also find significant decrease in lung tissue damage in histology level in asthma treatment group. This data makes the dupilumab wall. So the dupilumab demonstrated near perfect efficacy in near every aspect of HDM-induced asthma model. So it making it extremely challenging for novel candidates to achieve superiority results. So the question is how to overcome the dupilumab wall in asthma drug development. So it leads to the core of today's webinar, moving beyond the eosinophilic paradigm. While type 2 asthma is well served, there remains a massive white space for patients with non-type 2 asthma models who don't respond well to steroids or current biologics. So in our last section, we will move away from the standard OVA and HDM models and look at more complex, clinically relevant systems. Specifically, I will introduce our mixed granulocytic asthma model and neutrophil dominant asthma models. These platforms are designed to help you break through the dupilumab wall by targeting the pathway that currently therapies simply cannot reach. So in this section, we will parallel compare the type 2 and non-type 2 asthma model in the slides. And before we start the parallel comparison, firstly, I think I need to briefly introduce these non-type 2 asthma models. So firstly, I will quickly introduce the mixed granulocytic asthma model. So this model is the Alternaria alternata induced asthma model. So typically this model is activated by Alternaria alternata, and this model can be induced by the activation of innate lymphoid cell 2. So it has the characteristics of type 2 immunity activation. That's the key difference is that this is a fast-paced, short-term acute inflammation model. So which means it will also has the feature of neutrophil infiltration, making it a mixed granulocytic type with both eosinophil and neutrophil infiltration. So here is the study design of Alternaria alternata induced asthma model. So this is a short-term model. We will carry out all of the in vivo studies within 10 days. And in this study design, we evaluated the efficacy of anti-mouse interleukin-4 receptor alpha antibody as well as anti-interleukin 33 antibody. And the next model, non-type 2 asthma model I need to introduce is the neutrophil dominant asthma model. So this is the HDM-induced asthma model. So the key point in this model is that the challenge of LPS in this model switched the activation of the immune system from Th2 signaling to Th1 and Th17 signaling, so making it becomes a neutrophil dominant asthma model when the LPS stimulation is confirmed at high dosages level. So here in this model, we applied the challenge with HDM as well as LPS on the day 0, day 1, and day 2. And this data is for the sensitization. And on later days, in day 14, 15, 18, and 19, we will make HDM and LPS challenge. The whole in vivo study will take around 21 days, and we also evaluated the efficacy of anti-mouse interleukin-4 alpha antibody and anti-mouse interleukin 33 antibody. So that's the brief introduction of our mixed granulocytic asthma model and the neutrophil dominant asthma model. Now we will start the parallel comparisons between type 2 and non-type 2 asthma models. And I think this is also the key results of our today's webinar. So in this panel, I have listed three different asthma model. In the left panel is the classic type 2 asthma model. This is the HDM asthma model, and we apply the anti-mouse interleukin-4 RA as the test article. And in the right panel, I listed two different non-type 2 asthma model. The one is Alternaria alternata induced asthma model. This is the mixed type asthma model with the infiltration of both neutrophils and the eosinophils. And here the HDM plus LPS asthma model is the neutrophil dominant model. And the first aspects in parallel comparison is the body weight change. And here, basically, we can find that in non-type 2 asthma model like Alternaria alternata and HDM plus LPS induction model, it would result in slightly body weight loss, like 10% to 15%. And this phenomenon mainly results from the challenge of alternata alternaria as well as LPS, since these reagents has high immunogen activity, it will stimulate the immune system of mice model and finally results in the body weight loss. And the second aspect is the serum IgE levels. Basically, in both type 2 asthma model and the non-type 2 asthma model, we can find the secretion of mouse IgE in modeling group. Basically, there was a significant increase in modeling group compared to healthy control. And after the treatment of anti-mouse interleukin-4 receptor A antibody, we can find the significantly inhibition, basically the perfect inhibition in inhibiting serum IgE levels. So this is reasonable since the efficacy of interleukin-4 receptor antibody is mainly the inhibition of type 2 immunity, while serum IgE is a classic marker for the indications of activation of type 2 immunity. So it is easy to know that in both type 2 and non-type 2 asthma models, the efficacy of anti-mouse interleukin-4 RA persists across different asthma models in IgE inhibition. But when it comes to the immunotyping, we can find a completely different results. So when it comes to the facts analysis using BAF, we can find a completely different data. So here in type 2 asthma model, as we mentioned before, this is a eosinophil dominant model. And here we can find basically the number of eosinophils in BAF is around 150,000 in BAF, and while the number of neutrophils is around 6,000. So the number of neutrophil to eosinophil is around 1 to 20. It is a classic eosinophil dominant model. And after the interleukin-4 RA treatment, we can find the significant inhibition in eosinophils. And basically, the significant change and efficacy in mouse interleukin-4 RA would also lead to a decreasing trend in inhibiting neutrophils. But when it comes to non-type 2 asthma model, we can find different data. So firstly, in Alternaria alternata model, we can find the number of eosinophils is in modeling group is around 200,000, while the neutrophils is around around 100,000, and the ratio of neutrophils to eosinophils is around 2 to 1. So it is basically mixed the granulocytic asthma model. And in this model, although we can find a decreasing trend in anti-mouse interleukin-4 RA treatment group, we cannot find the significant decrease in this Alternaria alternata induced model. Besides, we can find the increase in neutrophils in anti-interleukin-4 receptor treatment group compared to the modeling group. So that's the first different data we found in Alternaria alternata induced model. And when it comes to HDM plus LPS model, we can find a completely different data. So in this model, the ratio of neutrophil to eosinophil is around 15 to 1. So it is a completely neutrophil dominant model. And the administration of anti-mouse interleukin-4 RA typically had no inhibition effect in eosinophils as well as neutrophils. And we can find even the increase uh, in eosinophils in anti-interleukin-4 RA treatment group. So in the BioFax results, we can find anti-mouse interleukin-4 RA failed to inhibit eosinophils in non-type 2 asthma models. So that's the first key findings we have talked about today. And based on these key findings, we can also find the similar results in the T cell and B cell in the BioFax analysis. And here we can find in type 2 asthma model, like HDM model, the administration of anti-mouse interleukin-4 RA significantly inhibited the total immune cell counts, as well as the T cell counts and B cell counts. But when it comes to the Alternaria alternata induced model, we can find there was no significant inhibition in anti-interleukin-4 RA treatment group in inhibiting the total immune cells, T cells, as well as B cells. And basically, we can find the similar results in HDM plus LPS induced asthma model. So here we can also find anti-mouse interleukin-4 RA failed to inhibit T cell and B cell in non-type 2 asthma models. And when it comes to the lung cytokines, we can also find the similar results. In the classic type 2 asthma model, like HDM asthma model, we can find a significant inhibition in the lung cytokines, like the mouse interleukin 5 and mouse interleukin 33. So we can find the significant inhibition in this type 2 immunity-related cytokines. But when it comes to non-type 2 asthma model, so firstly, in Alternaria alternata induced asthma model, we can find there was no significant inhibition in anti-interleukin-4 RA treatment group, while the administration of interleukin 33 can significantly inhibit the lung interleukin 5 levels compared to the modeling group in Alternaria alternata induced asthma model. And when it comes to HDM LPS induced asthma model, basically we can find no significant efficacy on both anti-interleukin-4 RA antibody and anti-interleukin 33 antibody treatment group. And in the serum cytokine levels, typically we can find the similar results. The anti-interleukin-4 RA treatment group showed no efficacy in inhibiting serum cytokines, while the administration of anti-interleukin 33 can significantly inhibit the interleukin 5 and the interleukin 13 in Alternaria alternata induced asthma model. So this results is consistent with the clinical progress we have talked, as we talked previously. And in the non-type 2 asthma model, typically we found no efficacy in anti-interleukin-4 RA antibody, while we can find the efficacy of anti-interleukin 33 in the aspects of inhibiting cytokines in the serum and the lung in mice. And when it comes to the HDM plus LPS model, typically this is a much more difficult model to find the efficacy of test articles. Basically, we found no positive results on both anti-interleukin-4 RA and anti-interleukin 33 treatment group. And when it comes to the histological examination, basically we can So firstly, we can find the inflammation as well as epithelial thickening in both asthma models. So firstly, we need to confirm the modeling success in both asthma models. So we can find in the type 2 asthma model as well as the non-type 2 asthma model, we can find the infiltration of inflammatory cells as well as the proliferation of epithelial cells. So which means the modeling of asthma model across type 2 and non-type 2 asthma is successful. And then we can find after interleukin for RA mouse antibody treatment, we can find the inhibition of inflammatory cells. So it will also reflect on the total asthma model score. So in the type 2 asthma model, we can find the significant decrease in total asthma score as well as airway wall thickness in interleukin-4 RA mouse antibody treatment group compared to the modeling group. And when it comes to non-type 2 asthma model, like in the Alternaria alternata induced asthma model, we can find both interleukin-4 RA antibody treatment and interleukin-33 antibody treatment can significantly inhibit the histology score. So which means both anti-interleukin for RA antibody and anti-interleukin 33 antibody can exert efficacy in inhibiting histologic level in this granulolytic mixed granulolytic asthma model. And when it comes to HDM-induced plus LPS-induced asthma model, in this neutrophil dominant model, typically, although we can find the significant decrease in anti-interleukin for RA antibodies, the overall efficacy of anti-interleukin for RA antibody is quite limited compared to the type 2 asthma model. So at last, I think we can summary all of the asthma models and the key findings in this slide. So typically for the eosinophil dominant asthma model, like the most classic OAA-induced asthma model, as well as HDM-induced asthma models, we can find significant secretion of serum IgE levels, and we can also find lots of abundant cytokine induction in lung. This is mainly related to the activation of type 2 immunity. And we can find basically the large proportion of both as well as lung eosinophils uh, in OAA or HDM-induced asthma model. And most importantly is that the dupilumab or anti-mouse interleukin-4 RA efficacy in this model is really good. And when it comes to Alternaria alternata-induced asthma model, it can also induce the serum IgE levels, and we can also find the secretion of interleukin-5 and interleukin-13 at serum and lung cytokine levels. By considering the acute characteristics of this model, we can find the infiltration of neutrophils as well as eosinophils. So in here, we can also find some of the efficacy in anti-mouse interleukin-4 RA. And when it comes to the mixed type of asthma model, like OAA plus chitin-induced asthma model, we can also find the serum IgE secretion and the cytokine secretion like interleukin-5, interleukin-13, interleukin-4, and interleukin-5. And here we can find basically the infiltration of both eosinophil and the neutrophil. And one last part, when it comes to the HDM plus LPS-induced asthma model, basically we can find the serum IgE secretion level is relatively low compared to this asthma model. And for the cytokine induction, we can only find a significant increase in interleukin-33 in the lung. And there are basically dominant expression of neutrophil in both, with very little expression of eosinophil. The lung histology score across these asthma model is relatively stable, which means the modeling phase for both of these asthma models are successful. While we can find in HDM plus LPS-induced asthma model, the efficacy of anti-mouse interleukin-4RA is quite limited, so making it challenging for the evaluation of dupilumab. So this model is actually a suitable model to break the dupilumab wall and to evaluate other test articles. And in the last part, I think I want to also take several minutes to talk about our autoimmune platform. And basically, besides asthma models, we have constructed basically a large proportion of autoimmune disease models. So the autoimmune disease platforms provides you with a one-stop services for in vivo as well as in vitro efficacy study. We deeply understand your needs and they continuously innovate and explore evaluation systems for innovative therapeutic modalities such as TCE, cell therapy, in vivo CAR T, small nucleic acids, and molecular glues. So in preclinical autoimmune disease models, our disease model covers 6 major systems, including respiratory system, like asthma we talked today, and skin system, like the atopic dermatitis, vitiligo, and the digestive system, like inflammatory bowel disease, and nervous system, like multiple sclerosis, and rheumatic disease, including SLE, the Sjogren's syndrome, the ankylosing spondylitis and like rheumatoid arthritis models, as well as kidney disease models, encompassing 42 indications and over 80 models. Distinctive models such as SLE and kidney disease have been utilized by multiple pharmaceutical companies to support IND filings and drug licensing and have been featured in oral presentations and posters at major international conferences. Guided by the principles of beyond execution, innovate solutions, we integrate cutting-edge therapeutics modalities with well-characterized disease models to deliver innovative mechanism-informed preclinical efficacy strategies to tailor to your needs. So I think that's all about today's webinar about asthma. And the next part, I think we can go into the Q&A session. Any questions?
Yeah, so we'll give everybody a chance to type their questions into the Q&A section on Zoom. There's a little button at the bottom of the screen. We see quite a few coming in. You pick one. I think we'll go with the most direct first. How do I choose the most suitable asthma model for a drug candidate? What goes into model selection?
Yeah, I see. I will switch the model here. So typically, I think firstly we need to divide the asthma model into 2 different types, the type 2 asthma model and the non-type 2 asthma model. So evaluating the test article in these 2 asthma models is completely different. So if you are aiming to evaluate test article in type 2 asthma models, then the main goal for the test article is to evaluate the efficacy in inhibiting type 2 immunity and compare the efficacy with dupilumab. So that's the key point. And if you want to test the test article in the non-type 2 asthma model, the key point is to aiming the efficacy in the non-type 2 asthma, mainly the mixed granulocytic asthma as well as neutrophil dominant asthma. So it is a completely different way compared to the type 2 asthma models. And basically, by using the non-type 2 asthma model, we can jump out the circle of dupilumab wall. So if you want to find the new efficacy of test article and try something very different from dupilumab, then I suggest to try out non-type 2 asthma model. Yeah, thank you.
Okay. Your data shows that anti-IL-4RA worked well in the HDM model, but not in type 2 models. Does that mean that the anti-IL-33 is a better target for these difficult to treat populations, or
Yeah, I see. I see. So that's the key point in non-type 2 asthma model. So as we said in the introduction in chapter 1, we said the clinical progress in interleukin 33 targeted antibodies. And here in our type 2, in our non-type 2 asthma model, and here especially the alternaria-induced asthma model. And yes, we can find the significant inhibition in anti-interleukin 33 treatment group. So this is consistent with other findings. So I think the anti-interleukin 33 is a good choice for non-type 2 asthma model, at least in the aspects of inhibiting these cytokines in lung and the serum, as well as inhibiting the lung damage in the aspects of inhibiting histology score. So this data has the evidence that the anti-interleukin 33 can has the significant uh, efficacy in inhibiting lung damage in histological levels. So, so I think the answer is yes. The anti-interleukin 33 is a promising target for non-type 2 asthma models. Well, basically we found very limited efficacy in anti-interleukin 4RA antibody treatment group. Yeah, thank you. Okay, I found one of the questions provided by Quan Chen. He said, "Would do you consider measuring lung function to evaluate the efficacy of the drug?" So I think so. Firstly, we have the capability to test the measuring the lung function. So this is not a problem for us. But when it comes to to the development of drugs. As for the preclinical studies, actually the lung functions, the efficacy of lung function is not compulsory or not the most important indexes in asthma. So in our opinion, the key points in asthma model, we need to focus on like the histology levels, the inhibition of like the inhibition of type 2 immunity or the inhibition of eosinophils or neutrophils. So that's the much more important aspects compared to lung function. Actually, in clinic and in preclinical studies, the lung function is not the main focus, and it is not the main indexes in asthma. Yeah.
maybe time for one or two more?
Yeah, and we found this question comes from Dan Sophie. So in dupilumab study and type 2 versus non-type 2 study included female mice, is any specific reason? So here typically in our research field, we apply female mice in all of these models. Basically, the reasons comes from the publications as well as the experience, like near 100 projects we have carried out in GemPharmatech. So I think the two reasons. One is the publications. Most of the publications apply female individuals in asthma model. And another is that we have carried out lots of asthma projects in GemPharmatech, and we found that female individuals do have the stability in different asthma models. Thank you.
Okay, we'll have Dr. Zhang choose one more. There's even more questions coming in.
Yeah.
So if he doesn't pick your question now, we'll definitely have someone follow up with you directly.
The Yeah, I saw one. Here is a question that where would you conduct this model? So typically, GemPharmatech is a global company. So we have subsidiaries across the worldwide. We have the facility. Our headquarters is in Nanjing. So typically these models will conduct in Nanjing, but we also have facilities in America and in other cities in China like Chengdu, Guangzhou, and Shanghai. So we can carry out these experiments on both in China or in America. Yeah, thank you.
Okay, so again, if we didn't get to your question, we will definitely have someone reach out to you directly. Thank you, everybody, for attending. Next month, we will continue this webinar series, and we hope to see you there. So thank you again for everybody for attending, and have a nice day.
Have a nice day.